Tonight we are pleased to have Phil Horner, Associate Professor in the UW Department of Neurosurgery and the Institute for Stem Cell and Regenerative Medicine. His presentation is called Stem Cells and Regeneration of the Spinal Cord, Practical Barriers and New Cell Technologies. There will be time for questions from the audience after his presentation. Please welcome Dr. Phil Horner.
I have three goals tonight. One is to give you a neurobiology lesson so that we'll all be certified neurobiologists by the end of the evening. And the second thing, my second goal is to tell you about, really define what a stem cell is and then tell you about different stem cell technologies. It's very confusing. I'm actually confused myself. There's so many different kinds of cell types now you hear about in the news. So I'm going to tell you about a few of them. We won't be able to talk about all of them. I'm going to tell you about a few of them, and one in particular that I'm very excited about. And then, if we can make it that far, if there aren't too many questions, I'm going to show you really a practical neurobiology experiment, which I think has great relevance to the treatment of spinal cord injury as it pertains to or the transplantation of stem cells. And I'm going to highlight one of the, I think, biggest barriers we're facing, and that is most trials or preclinical models that people have been focusing on with stem cells have been directed towards acute spinal cord injury and not subacute or chronic spinal cord injury, which is what most of us are interested in here. So, okay. So I show this because how many people think that stem cells are a revolution? This is from Time magazine. Would you characterize it as a revolution? I would have a tendency to agree with it. It's what I work on, so I have to get excited about it. But I think that this is, stem cells, the concept of stem cells has reached a crescendo of excitement. And in recent times, that sort of sense of excitement has actually started to hit practical reality, that there are some real instances where stem cells may be beneficial in a variety of organ systems. And we're going to talk about primarily the spinal cord today, but stem cells really do seem to be a coming revolution in terms of their capacity to treat disease. I don't know if they say it could save your life. Stem cells could save your life. It kind of gives the impression you might be carrying around a vial of stem cells with you, like you would carry around a vaccine or whatever, something that you might need at any moment. And this is maybe not the case. And what we're going to talk about tonight is some of the barriers for using stem cells in a practical sense. So, Neurobiology 101. We'll all start here. Some of you probably know this really well, and for those of you who don't, we'll just bring everyone up to speed on a little bit about the nervous system, so you'll be certified neurobiologists. So how many of you know what these cells are? Anybody can tell me what that is? This is a cell type in the brain and spinal cord. They're neurons, that's right. And what's unique about neurons in the brain? They do have one very unique feature. Does anybody know what that is? It separates them from other cell types in the brain. They are excitable cells. In other words, they can send signals to one another. The only cells in the brain that send signals from one to the other. And this little caricature, this is a neuron here, there's a neuron here, and you see all these processes going off. And these little glowing areas are supposed to simulate the idea of a message that comes from the cell, and it's shooting down its process and going to another cell. So these are the neurons of the brain, and they're also in the spinal cord. And neurons in the brain connect the cells of the spinal cord, and these two have to communicate. When there's a spinal cord injury, you disrupt the communication by basically breaking some of these little processes. And the goal for restoration of spinal cord function is to restore those connections. That's really the ultimate goal. And I can tell you that we've got a ways to go to that towards that goal. And one of the reasons for that is because of the complexity of the nervous system. And I'll show you that in a moment. So what other cell types are in the brain and spinal cord? We need to know these so that when we talk about stem cells, we need to know why we're making different kinds of stem cells. So there's another type of cell. The rest of the brain is made up of a type of cell called glia. This is a term that used to be referred to as the glue of the brain. Does anybody know what glial cells are? Supporting cells, that's right. So in the really old days, back in early Greek physicians and so forth, they used to think of these just glued the brain together, just kind of held it together. And then now we know that they're actually provide an important support function. There's various kinds of glial cells. There's oligodendrocytes. This is a type of cell that makes insulation around those nerve processes I just showed you, nerve processes called axons. So there's the nerve process, and this is a oligodendrocyte, and it forms an insulation. So just like an electrical wire Every nerve cell really needs insulation. And if you lose that insulation, which you often do in spinal cord injury, wires get crossed, signals get messed up, and you don't get good conduction. So that's one type of cell. The other type of cell, which may be a little hard to see with the lights at this level, but are a type of cell called astrocytes. And people often refer to these as having a nursing function for neurons. So astrocytes, I hope you can see it, but you can see the little blue processes all over the place here. And the big red blobs are neurons. And I just want you to notice that these astrocytes, these special types of glial cells, just keep their fingers and they run their processes all over neurons. They're essentially hugging the neurons. And they provide a very important nursing function. So neurons are busy creating signals, and glial cells do something that's very it's essential for function. They basically provide all the nutrients to the neuron, and they take out the garbage for the neuron. They create the environment around the neuron, keeping it healthy for function. So the neurons have an intimate relationship with glia. They're really essential. And this intimate relationship between the neuron and the glial cell becomes disrupted in spinal cord injury, and it's going to be a key element in the talk I'm going to give to you tonight, which is what happens to glial cells after injury, and how do stem cells, when we transplant them, for example, how do they interact with these glial cells? What are the changes that occur there? So there is a very famous scientist named Santiago Ramon y Cajal. He's sort of our Mendel or other famous scientists. Neurobiologists think he's really the father of modern neurobiology. And not much has changed since Ramon y Cajal's time. Ramon y Cajal, what he did is, his father wanted him to be a doctor, but he really wanted to be an artist. And back in those days, when your father told you to be a doctor, you just did it. So that's what Ramon y Cajal did. But he was passionate about the structure of the nervous system. And you can see a picture of him here looking through the microscope. And what he did is he drew the nervous system and drew incredible, exquisite theories about how the nervous system is built. And he actually predicted the different types of glial cells that are there, how neurons function, and he created something called the neuronal doctrine, and which is really true. This was in 1911. He developed this theory, and it's proven true. Very little has changed in the time since Ramon y Cajal. And this is a drawing that he made of a spinal cord, and you can see all the little fibers in there. Those are all the little nerves processes that are in the spinal cord. And one can imagine that this is, obviously, just by looking at it, it's very complicated. All of the information there and the processing that occurs between these neural processes. And this is what's disrupted after a spinal cord injury. So we have two goals. We have to try to get nerve to regrow from the brain back towards the spinal cord. But the other thing is we've got to make sure this structure, this complicated structure, is recapitulated in such a way that you have function. And there's good news. And that when you see that, and Ramon y Cajal concluded that it couldn't be done. So he said that once a nerve is injured, there's really no hope that this is that the endings of the nerves become frustrated, the end bulbs become frustrated, and they can never regrow. He said everything can die. In fact, what Ramon y Cajal says, everything can die, but nothing can regrow. Now, he was a wonderful neuroscientist and neuroanatomist, and he was absolutely right on almost everything except for that last point. And I'll tell you why. And that is because we know that at the advent of stem cell biology tells us that we can create new spinal cords, and also there are model systems in which new spinal cords are created in lower species. And we'll talk about that too. Okay, so what happens in spinal cord injury? You have this is just an MRI from a spinal cord injury, and you can see there's a central injury here. The most common type of injury is a contusion or a bruising of the spinal cord, and there's injury there. So we have disruption of the signals, the axonal processes or the neuronal processes up and down the spinal cord. Now, we know that there are stem cells that exist in the adult nervous system. We know there's stem cells there's stem cells in the embryo and so forth. We'll talk about them. We know there are stem cells in your spinal cord and in your brain. We're going to talk about those in a minute. So the question is really when we think about stem cell biology, we're thinking about, can you make stem cells, turn them into replacement parts for those individual cell types we just talked about? So we want to make new neurons, but we can't just make new neurons. We have to make what other kind of cells we have to make? We have to make new glial cells too. And that's what this diagram is showing, is that we want to take stem cells and we want to teach them to make the individual cellular components that need to be replaced. So that's the goal, and I just showed you how complex the spinal cord is. And we're going to start now. You're all neurobiologists because you know the different cell types, you know who Ramon y Cajal is, and you know that the spinal cord's complicated. So congratulations. That's good. All right, so what defines a stem cell? We're going to talk about stem cells now and different types of stem cells. Can somebody throw out a definition of a stem cell? You can program them to do this. So that's a function of a stem cell. You can, and that's what we're trying to do. But if you had to make an operational definition, so if you had so for example, you have a blob of cells. How do you know those are stem cells? What qualities should they elicit or exhibit? They can become any kind of cell. Okay, that's one criteria. There's two essential criteria. We're halfway there. Anybody? And they can reproduce. So they are self-renewing. Very good. And some people call them multipotent, which means they can become different cell types. And then I add this, that their fate, what they become, is actually determined by its neighbors or its niche. So this is the standard definition of a stem cell. It should be self-renewing, and it's multipotent. And something we know as of late is that really the fate of stem cells is determined by what they're next to. Stem cells require a partner in their program to what they were going to become. And we're going to talk a lot about what the partner is in the injured spinal cord. So let's get more complicated. So if we that was great. So here's a little bit more deeper terminology. And it's helpful because when you read the paper, or even when you read articles, you hear all kinds of terms: pluripotent, multipotent, self-renewing, asymmetric cell division, all kinds of things. And this is just, and you'll have this in your handout, this is just a little bit more detail on what some of the jargon is surrounding stem cells. So I like to think of stem cells, a true stem cell, the ultimate stem cell, really is immortal. And what I mean by that is that the test of whether a cell is a stem cell, it should survive. If it's truly a stem cell, it should survive the entire lifespan of the organism, because only a stem cell can do that. So a stem cell is there when you're born and should be there when you die if it's truly a stem cell. It has infinite the ability to replicate itself infinitely, and that lends itself immortality too. In other words, it can produce an exact copy of itself, and that's a form of immortality. It gives rise, so the other function that this immortality has is that it gives rise to other cell types. And these cell types lose their stemness to become progeny, if you will. They're progeny that are committed and produce different cell types in the body. So a stem cell really is able to survive the entire lifespan, can replicate as often as it wants. It can create an exact duplicate of itself. And that's unlike any other cell in the body, because we all know as we're aging, every minute, our cells are aging. But stem cells do not age. They don't exhibit the same characteristics as any other cell in the body. And we'll prove that to you a little bit later. And they have this wonderful ability to generate children or progeny that can produce cell types that are important for repair or homeostasis. The other quality, so another way to broaden these terms out, is that they're highly plastic. Now, different kinds of stem cells have different kinds of plastic properties. The common terms that are given are multipotent, and I define multipotent as a cell that can produce more than one cell type. So if you're multipotent, it means you don't just make a skin cell. You might make skin and blood vessel cell, but you don't make just one thing. That's true multipotency. The next term that you see often used is pluripotent. There are multipotent stem cells and there are pluripotent stem cells. Pluripotent stem cells can produce the phenotype of the type of cell of more than one organ system. And that's a good sort of operational definition. If you hear a stem cell is pluripotent, it implies that it can make heart and it can make lung, or it can make bone and it can make kidney, or something like this. It can make multiple organ system type cells. And the ultimate stem cell is the totipotent stem cell, and there's only one of those, and that's the fertilized egg, or the zygote. Because a totipotent stem cell, totipotency implies that it can create a whole new organism. So when you read about a totipotent stem cell, that's really what people should be talking about. It should be really the fertilized egg. It's the only cell that can do that. All right. So I'll be using some of those terms, and now you know them. But if you forget them, you can ask me again as we go, or you can challenge me if I use them wrong, because I probably will. All right, so what types of stem cells are there? There are so-called embryonic stem cells. And embryonic stem cells are derived from the blastocyst. We'll talk about that in a moment. They are pluripotent, and they're probably the most popular stem cell in terms of public interest, I would say, and scientific interest even too. So, but these are pluripotent. So what does that mean? What is an embryonic stem cell if it's pluripotent? More than one organ system. But can it make a new organ? I mean, a new organism? No. Very good. If we had you guys around the last few years with all this debate going on, we'd have been in great shape. So what about fetal stem cells? So the fetal stem cells are derived from the post-embryo, I call it. So anything after the embryo, which is in humans about greater than 8 weeks. So if you have anything older than 8 weeks, that's called a fetal stem cell or fetal progenitor. And these are typically lineage-specific cells or lineage-specific progenitors, meaning that they like to produce a specific type of cell. They're more restricted. They're not pluripotent, they're multipotent. There are perinatal stem cells or perinatal. I'm using the term progenitor and stem cell a little bit ubiquitously, but I'm doing that on purpose. Stem cells and progenitors are similar in terms of defining these types of cells. There's perinatal stem cells. These are derived from the early postnatal period, and they tend to be tissue-specific and developmentally specific, so the time postnatal of what they're going to produce. And the last type of cell is really an adult stem cell. And these are multipotent stem cells, but they're derived from adult tissues, and they almost exclusively turn out to be tissue-specific. So they're multipotent. They're not pluripotent, they're multipotent. All right? With so many stem cells, it's hard to keep them all straight. So let's go through embryonic. So does anybody know where the embryonic stem cell comes from? Larry, from the embryo. I went to my father-in-law for the answer on that one because I knew he would know. So, and the embryo, it's funny because in embryology, when I remember embryology, this is what I specifically remember, but embryo, as I told you, is that first 8-week period. And the only time you can get embryonic stem cells is when the embryo forms the specialized structure called the blastocyst, which is here. And there's a little area inside the blastocyst called the inner cell mass. They suck that out with a pipette, and they put it in a dish. And this is a pluripotent stem cell. And that means that it can make neural cells, it can make heart muscle cells, it can make blood cells, it can make any cell you want from the body. And so this is the embryonic stem cell. And we'll talk a little bit more about them. Any questions about them? So that's where they come from, that little blastocyst. All right? So you hear all the good stuff about embryonic stem cells, and when you hear you can make any cell in the body, that sounds fantastic. But there's some problems.
How long does the blastocyst last?
Just a few days. So this is a very short period of time when you can take cells out of there before it moves on. So what are some of the troubles with embryonic stem cells? So why are they not a panacea? And this is some of the problems with them, and it gets to why other stem cells may be important. So one problem with them is they're pluripotent, and you'll say, well, Phil, that's one of their best qualities, isn't it? And why would that be a problem? Because if they can make any cell type in the body, and if you want them to make a specific cell type, say a heart cell, or you want them to make a brain cell, you've got to make sure you can control them so they only make heart or brain, because you don't want brain in the heart and you don't want heart in the brain, right? Okay. So that's one problem that people are working really hard. It can be hard to control proliferation. In other words, it can be hard to control how many times an embryonic stem cell divides. And one of the reasons for that is they want to make an embryo. So we actually have to trick them to prevent them from dividing too much. If you want them to make a few brain cells in your brain, you don't want to make them a whole brain, it's a little bit hard to control. The other thing is that cells do not understand, embryonic stem cells, they don't really understand the adult environment. An embryonic stem cell has never seen the adult. It doesn't make the molecules to interact with the adult tissue. So an embryonic stem cell can make a heart cell, but if you take that embryonic stem cell and you jam it in the heart, it doesn't know what it's doing there because it hasn't gone through the developmental process. It just doesn't know how to interact with the adult environment. And that's one of the hurdles that people are struggling with. The other problem with embryonic stem cells in general is that they would theoretically will require immune suppression because they are going to be from they are not from you. So, and the other thing is that it's hard to make therapeutic clones. I'll talk about that both ethically and practically. So one way to get around immune suppression would be make therapeutic clones. And how many of you heard of therapeutic cloning? That was a big deal for a while, yeah? Therapeutic cloning. So therapeutic cloning, the idea there is that you would take an embryonic stem cell. Well, you'd take an oocyte from a donor, and then you take the nucleus out of that oocyte, you suck it out, and then you take an adult cell from a donor, you take its nucleus, and you put it in that oocyte. So now it has the nucleus of the donor and the oocyte of the cell of the oocyte donor, and then you put some magic electrical shocks and chemicals and what have you there, and it makes a new blastocyst. And then from that blastocyst, you can suck out, remember that early embryo, you can suck out those cells and you can make embryonic stem cells that theoretically have the nucleus from the adult donor, from this donor. So if this person had a spinal cord injury, for example, you could take their cells and make their own perfectly matched embryonic stem cells. Now the problem with that is that it's very hard to do this efficiently. And how many people remember the Korean cloning, therapeutic cloning? So everyone got really excited about that because they were getting very good results with therapeutic cloning. They were claiming that they needed as few as 60 oocytes to get a functional clone, but we know that turned out to be not the case. This turned out to be a difficult process, not a very efficient process, and that brings into account a lot of ethical issues and, of course, practical issues in trying to create clones for each patient. And now we know that Hwang Woo-suk, who's the author on that, he's on trial for fraud and embezzlement. Okay, so a new technology that's out there that might replace embryonic stem cell technology is what was just brought up, and that's something called the induced pluripotent stem cell. And what is an induced pluripotent stem cell? This is from an article by Janet Rosette, who called it the magic brew. And this is really, this was done simultaneously by a group in Japan and a group in the U.S., and it's really kind of like science fiction. You don't think this would really work. And what they did is they looked at they took an adult cell from a mouse and a skin cell, they put it in a dish. And then they asked, separately, these two groups asked, what are some of the genes, some of the master genes that control stemness, if you will, from an embryonic stem cell. So what makes an embryonic stem cell an embryonic stem cell? And they picked several of these genes. In this case, they took 4 of those genes. And they asked, what if you could just take those 4 genes and put them in an adult cell? Would it behave like an embryonic cell? This is the kind of research you'd never get funded from the National Institutes of Health or anywhere else I know, because it just kind of sounds crazy. They only want you to focus on one thing, and if you say, I'm going to put 4, 6, 8 genes in there and see what happens, no one would pay for this. But both of these groups did this simultaneously. This is just an example of the genes they put in. So they used technologies where they could force the production of these genes in adult skin fibroblasts from mice. And then they have a way of selecting for the cells that have all 4 genes in them. And they propagate those cells, they grow them, and they have the qualities of the embryonic stem cell. And this is really a revelation because that's going to be a lot easier than the therapeutic cloning, you can imagine, if it's really true. I could take a skin cell from you, jam 4 genes in there, and suddenly I have an embryonic stem cell from you. So what was one of the true tests of whether something is a pluripotent stem cell? Do you remember what some of the qualities were? There was one that had to be self-renewing. What was the other one? Multipotent. What else? Live the life of the organism. So the true test, and reviewers didn't believe this at all, and I wouldn't have either probably if I reviewed it, just seemed kind of crazy. So they forced these groups to say, well, if this is really a stem cell, it should be able to survive the entire life of the organism and come out the other end, be a stem cell. And they actually proved that. So that's the way they proved that this technology is real. So they made these induced pluripotent stem cells. They made blastocysts from them, these embryos, and they took those cells and they injected them into a mouse. So they made what they call a chimeric mouse. It has some of the induced pluripotent stem cells, the yellow cells here, and some not. And then they mated that with a normal mouse, and they could follow it through the germline. And there was some mice that have completely derived from the induced pluripotent stem cell. So the stem cell then can be injected, it survives the bloodline, and produces a whole new, or can contribute to the production of a whole new mouse. So this tells you that this is really probably a stem cell, right? I mean, it's a true stem cell. There are other tests you might ask is whether or not they're truly multipotent. That was brought up, whether they're pluripotent or multipotent. So this is actually data from our laboratory, and we've taken induced pluripotent stem cells. Now these are from human. These are from an adult male human skin biopsy, where we put the 4 genes in there. And normally, skin cells look like this. They're flat and long. And in this case, what we did is we put the genes in, and when you put the genes in, they immediately begin to look like this, which is really like a little embryo. They start looking like a little embryo. And they also make these brightly colored cells, or just different genes that are normally found within an embryonic stem cell. So they make those genes. And through a variety of processes where we push the cells down different pathways, we can get them to produce brain cells. So the green color you see here, the green is a protein that's made by neurons in the brain. So here they're making neurons, and the red you see here is a protein that's made by glia in the brain. So this shows you that we can take an adult human and skin cell and turn it into brain cells, which is really a revolution conceptually for the treatment, for the ability to create an autologous cell that won't need immune suppression. Any questions on that? So the question is, are rat and human similar in terms of their spinal cord? And I have, I guess, 2 points on that. One is I think they're anatomically very similar. The cell types are the same. The structure is the same. There's one fundamental difference with rodents in particular, and that rodents are quadrupedal. And rodents do a lot of their walking, and this is important when you interpret the literature and you hear people talking about how the rats are walking in certain therapies. A lot of walking in a rodent is what we call spinally mediated. In other words, it comes from the spine. There's not a lot of brain involved. Cerebellum and other parts of the brain are not that involved in walking. That's because they have 4 legs, and the 4 legs can communicate, and they do communicate with each other through the nervous system, through the spinal cord. So that's a fundamental difference. And so but they're very similar anatomically. It's a very important question. All the same cell types are there. So we figure if you can get a nerve to grow down a rat spinal cord, you can probably get it to grow in a human spinal cord. But the only caution I add on that, and I think it's a really important one, is that if you get a rat to walk, it doesn't necessarily mean you get a human to walk, because it's much more complicated to be bipedal. You need a lot more information from the brain. So is there an issue with the lifespan? That was the question. Is there an issue that the rodents live a shorter lifespan? There might be. Everything's kind of accelerated also in rodents. Their reaction to injury, their immune system works faster. So things are kind of accelerated too. But I think that could be important. But the big problem is we don't really model chronic injury in rodents very much at all. We mostly just do acute modeling. And so for that, it's probably fine. There are other systems that people have used. People are working on primates sort of as a final stage, and other organ systems that might be better. Okay, so now we're going to talk about adult neural stem cells. So we talked about embryonic stem cells. We talked about these induced pluripotent stem cells. I'm going to skip fetal stem cells for now. I'm going to talk about adult neural stem cells while they're important, and then we'll come back to the concept of transplanting these stem cells into an injured cord and talk about some of the roadblocks there. So this is a study by Steve Goldman, who was one of the first to show you could take, when a patient has epilepsy, sometimes they'll remove part of the brain that's damaged from the epilepsy. And Steve took those biopsies from humans, put them in a dish, and he was able to show you could make a adult neural stem cell. And that's what you see here. These balls are adult balls of neural stem cells. And Steve showed that they can make neurons and different types of glial cells. And this wasn't the first time it had been shown, but it was really one of the first times it had been shown in human. And this was kind of surprising because, again, it suggests that humans have stem cells. And so it begs the question, why, if you have a stroke or injury either to the brain or spinal cord, why isn't there more gross replacement of lost cells? Does anybody know what the adult neural stem cell does in your brain? I mean, so everybody in this room has adult neural stem cells in their brains. Do you have any idea what they do on a daily basis? Just hanging out. They can't be doing too much, right? We know they don't create, gross repair of the brain, but they are very busy in your brain, and I'm going to tell you a little bit about that. So this is an area of your brain called the hippocampus, and this is a slide of the brain. It's called the hippocampus because it looks a little bit like a seahorse. And this is an important area of the brain. It's considered the gateway for learning and memory. So all new memories have to come through this area of your brain. They get processed there and then get transferred to long-term memory. Okay? Pretty critical. If you were to get a lesion or an injury just at the hippocampus, you really would lose the ability to store memories from that point. So this area is so important. I always like to point out it's so important. It has its own journal, hippocampus. That's when you know your brain structure is really important because you have your own journal. Okay, so this area of the brain, turns out, here in a human brain, we're looking at, is an area where these adult stem cells make new neurons. So this area of the brain called the hippocampus, your stem cells divide. They produce stem cells that divide and produce immature neurons. These immature neurons migrate up into a layer of neurons and produce fully differentiated neurons in your hippocampus. And there's good data to suggest that this happens all the time. I'm going to give you some numbers here to hopefully impress you. But this suggests that so we know you have an adult neural stem cell, and we know it won't repair you completely if you have a spinal cord injury, but they're busy in your brain in certain structures, and this should give you hope in thinking that if it can do this, maybe it can do more of what we need for repair. So this has also been found in humans. This is an article in Nature Medicine, which proved that humans have this process. What I showed you before was a rodent. So these are some of the numbers, and there's some information here that I think is surprising. So if you look at, this is complicated data, but if you look at some of the key things. So how many newborn neurons are there in this structure over your lifetime? It's a considerable amount of cells. So in rodents anyway, it's 9,000 new cells are born every day in that structure. And that's in a rat, and they have a smaller hippocampus. So in human, this is probably pretty extensive. And what that adds up to is 3.3% of all the neurons in that structure are replaced every month. And that means that the hippocampus that you're born with is not the one you die with, probably, right? It's probably replaced considerably. And this is really a revolution in thinking, because it means that certain parts of your brain are highly plastic, and stem cells can replace the neurons there, and they do on a daily basis. If you block this process, what do you think happens to learning and memory? You lose it. Exactly. That's right. So this is key. This is the gateway for learning and memory, and there's something about the birth of newborn neurons, these young immature neurons, that's critical for new memories to be coming through this structure. So we're marching further into asking how adult neural stem cells behave in the brain, and then why this is important for spinal cord injury will become apparent in a minute. Because you have stem cells in your spinal cord, I told you that. So what do you think can influence this process? If the birth of newborn neurons is important for the acquisition of memory, do you think anything might stimulate that? And key question, the fundamental question everybody likes to ask, is it nature or nurture? So are you born with this certain level of this process? Or can you manipulate that process by certain things? So how many people think it's nature? You raise your hand. No one thinks it's nature. Couple. Or both. Everyone say both. Anybody to think it's nurture? It's got to be both. All right, so this too smart a group. So let's take a look. There's evidence for both. I'm just going to review it quickly. So this is the evidence for nature. It's hard to do nature in rodents, but this is a study done by Gerard Kemperman a long time ago. And what he did is he took different strains of mice, and scientists use these different strains of mice for different reasons, and he got a bunch of them together, and he tested them to see which ones were smart and which ones weren't so smart. And then he measured the birth rate of newborn neurons in their brains. And he found that the really smart mice, in this case, this is a strain called C57, and this white bar shows you had a very high rate of stem cell birth in its brain, and it was this particular strain of mice is much smarter than this 129, for example, and he showed that there's a good correlation between the birth of newborn neurons and the intelligence of these mice. Now, that's good evidence for nurture. Hard to do these studies in humans. We can't do them yet, so we don't know if it's true. But so that might be the bad news. You're born with a certain rate. It certainly implies that you're born with a certain rate of neurogenesis, and that may correlate to some degree with your intelligence. But there is good evidence for nurture. And so what Geert did is he took those same strains of mice and he put them in an enriched environment. So this is an enriched environment for a mouse. So this is a normal mouse cage here, and this is a mouse cage with a running wheel, and this is a so-called rich environment. So all the different tunnels, they have more friends to hang out with, running wheels, and so forth. And that's their enriched environment. They love it in there, incredibly active. And this study, what Geert found was that here, if you just look at this is the quantitation of that data. The red bar is the unenriched animal, the number of newborn neurons in its brain, and the blue is the ones that live in the enriched environment. So he could double the rate of newborn neurons by enriching the environment of these mice. And he could take the mice that aren't as smart, put them in enriched environment, and they would nearly catch up with the naturally smart mice. This study was fascinating because it did suggest that there's a physical change in your brain when you have enrichment, and it's tied to the birth of new cells. There's also evidence that exercise alone might be sufficient for this effect, and that is. Depending upon how you look at it, that's good or bad. So this is a study in which Henry van Praag put mice in a variety of different environments. The first bar you see here is just normal mice in a normal cage. This was in this is called so-called learner mice, and that's where she would put them in a behavioral test just to teach them something new. And then there were swimmers, so mice that got swimming, very stressful for a mouse. And then there were runner mice, and running on a running wheel for a mouse is like the happiest they can be. They run 5 kilometers per night on these wheels when you put tachometers on. They're pretty amazing. And then these are the mice that are in the enriched environment. So this is the number of dividing stem cells in their brain, and then this is the number of dividing stem cells that became new neurons. And you can see both runners and the mice in the enriched environment gain a lot more new neurons. So this showed that maybe, for a mouse anyway, being on a running wheel was sufficient to really stimulate both their learning and memory capacity, but also the birth of newborn neurons in their brain. There's very strong correlates with this in spinal cord repair and physical rehabilitation, because a lot of the same factors that regulate this process are upregulated in the spinal cord during physical exercise. And I won't have time to talk about that, but there's a lot of information on that if anyone's interested. Okay, so the net effect of this research, has it really impacted humans yet? I don't know, but the mice, they all require these things now. All right. Okay, so there's really an expanding list now. There's a variety of factors that influence this process, from running to enriched environment to diet, variety of things. I won't go through the list. This is a short list. There's many more. But it turns out that really stimulating this process is very sensitive. It's downregulated by alcohol. It's downregulated by stress, by mood. It is positively regulated by a variety of growth factors, diet, and activity. Okay, so why did I tell you all that? I mean, I told you that because it's a cool story, but it does have great relevance to the spinal cord repair. So in sort of summary of the first part of the talk, there's many types of stem cells, I told you, and they all have their advantages and they all have their disadvantages. And the question, I think, where we are as a field is that we've kind of gotten really excited about stem cells from that Time article and that cover article. It's a revolution. And the thing that we are just realizing that we have to do more of is we have to understand what the goal is for these stem cells. We can make them into any kind of spinal cord stem cell or any kind of cell that might be found in the spinal cord, but what kind of cell do we really need to repair the spinal cord? And believe it or not, that's not really clear yet. So we need to define what the stem cell needs to become in order to repair the spinal cord. And then we also need to know how the environment of the spinal cord changes after injury. And I'm going to show you, I think, dramatic evidence which shows that over time after injury, the environment changes dramatically. And that we can't, for the design of clinical trials, for example, the time after injury is going to have a big effect on how stem cells interact with the injured cord. Okay, so stem cell behavior is the what I like to refer to as the stem cells depend upon their neighborhood to determine what they're going to become. And the stem cells are regulated by both internal cues and external cues. So if there's any other sort of fundamental, and I hinted to this earlier, fundamental thing you should know about stem cells is that most stem cells, almost every stem cell that's been studied, and the stem cell exhibited here, almost all stem cells have a partner cell. Stem cells don't do well in isolation. They have to have a partner cell. And that partner cell they're connected to by a very intimate interaction, specific proteins, lock and key, and they can actually transfer information between that so-called junction. And this is universal. All stem cells so far has been studied have a stem cell and a partner cell. And it's this interaction between some partner cell that allows the cell to be a stem cell. So if you take a stem cell out of that environment, it loses its stemness. It loses its stemness. It requires this interaction. And that interaction is different if you're an embryo, or if you're a fetus, or if you're a heart, or you're a brain. That interaction and the partner cell is always different. And so we're just beginning to understand what the partner cell is. And we're going to talk about spinal cord. So what are the partner cells? And what are the signals? What are the signals that the stem cell will encounter when it partners up, when we transplant them into the spinal cord? So the partner cell really tells the stem cell when to become something else. And when it does that, the stem cell divides, and it makes a copy of itself, an exact replica of itself, and then it makes one copy that is no longer a stem cell. And that cell is called the daughter cell, and it goes off to become a neuron or a glia or something else. And this process is also important to understand how stem cells can be used clinically. So I just told you that's a stem cell niche. There are examples of regenerative niches. So this is now a proof to you that Ramon y Cajal is wrong. Remember what Ramon y Cajal said, everything could die, nothing can regrow. Right? Okay. So this is Ramon y Cajal, for whatever reason, I think he just wasn't caught up on the starfish literature and he didn't know this data. But we can't. He was pretty good in everything else. So if you look at starfish, there are species that regrow, and there are species that regrow relatively complicated nervous systems, and I'll show you that in a moment. This is a starfish, and most of you know that if you cut off the leg of starfish, it'll regrow a new leg. But what's really interesting is that leg can regrow a new starfish if cut off at the right point. And that's remarkable because it tells you all the information for the starfish is contained within that little niche at the end of the arm. So there is a niche there that partners with the stem cell and instructs it, both in the polarity, where the arm should go, and the size of the arm, and everything is all contained within that niche. It's not in some magical factor that distributes throughout the starfish. It's actually in one zone. There are other species that regenerate, and more pertinent to what we're interested in today. These are salamanders, axolotls, and you can buy them at the pet store. They can regenerate their limbs. So if you cut off the arm of an axolotl, it produces a limb bud. That limb bud slowly generates a small arm, which becomes a fully functional limb. And continue with the same idea of a niche. Now, what's cool about this salamander is if you take that limb bud, so if you take this limb bud off here, and you can, when it just gets initiated, you can take it and you can put it on the animal's forehead or on its back, and it'll make a fully functional limb most of the time there. So what does that tell you? Where's the niche for regeneration? It's right in the end. It's not in some magic body plan. So this thing gets organized and it makes a new limb. I'm sorry? So it is in. So he's asking, is it on the end or is it in the cell? And it is the environment of the cells. It's the cells that are in that end that begin to polarize. And it starts with a cell. That's right. It starts with a cell. But there's a stem cell and there's a partner cell. There has to be two cells to make this process happen. Okay. But it starts with just a cell. So salamanders can also regenerate their spinal cord. So if you cut the spinal cord of a salamander, so here's one end of this salamander's spinal cord, here's another end, and there's a gap here. You wait a few days, it fills in with stem cells. And it regenerates from the head back, just produces a new spinal cord. So Ramon y Cajal would have been shocked by that, right? Because that's pretty impressive. It's a complicated structure, and the salamanders have a real spinal cord. It won't go all the way. It won't go all the way from the brain. You have to cut it somewhere in the spinal cord, and it'll go. But it regenerates from the cut end all the way to the tail. And this is what it looks like practically. These brown cells you see here, every brown cell is a cell is newly divided. It's a newly divided stem cell. This is just 24 hours after we cut the salamander's spinal cord. And these red cells you see here, those are new neurons. So the salamander, within a day, produces a new embryonic type spinal cord, and the first thing it does is start producing neurons. So that's important to know. The first thing it gets set about is just producing a new spinal cord. There's no messing around. And salamanders, can run around after this quite normally. Okay, so what is the mammalian response? What is our response, the human mammalian response to spinal cord injury? We're going to compare and contrast it to what we just saw in the salamander. Well, this is a caricature of what happens in the human spinal cord. So after spinal cord injury, there's a bruising in the middle of the spinal cord, typical type of injury, and blood products and inflammation leak into the spinal cord, and there's a hole. And this is going to be important later. This hole in the spinal cord never gets filled in with new neural tissue. It becomes a devoid area of neural tissue. And then around that hole, there is a scar, a classical scar, just like you would have in other parts of the body. And this never reduces. For whatever reason in mammals, it never reduces. In humans, in 30 years after an injury, anything happens, it just gets bigger. And then out here we have the so-called spared spinal cord. And there's a variety of pathologies there. Okay, so we remember what happened in the salamander. You get very quickly new stem cells and new neurons are being produced. This is what the mammalian spinal cord does. So the exciting part is that mammals, surprisingly, and this is just discovered in the last few years, that we do start to make a new sort of embryonic structure. This is all the green cells are new stem cells dividing right in the middle of the mammalian spinal cord. So that happens in mammals. But obviously we don't get a new spinal cord. We wouldn't be here tonight. So what's the big difference? The big difference is that instead of going on to produce neurons, as you see here, mammals produce only glia. And this is a type of glial cell here that our lab studies. So they go about making glia. And that might not be a bad thing, but the bad part of this whole process in mammals is that the type of glia they produce are so-called scar-type glia. So they contribute to the production of a scar. Very, very good. So this is an image from our lab where we reconstructed stem cells that were born one day after injury, and we looked at them a week later. And all the red substance you see here, that's a marker of scar-type cells in the spinal cord, and that's what they're making. These are the newborn stem cells. So our stem cells make a scar. If you look at this as more of a caricature, this is really the crux of the problem for repair of the spinal cord. So these are cut ends of axons. And these are these newly generated scar cells, and people have shown that there's all kinds of factors that are made by these scar cells that block the regrowth of those cut nerves. So that's the mammalian response. We don't know why it's like that. Why, I can talk about that for hours. Why mammals might have evolved that way, but we don't really know. But the question is, if we know a little bit about stem cell biology, maybe we can fix it, right? Maybe we can become a little bit more salamander-like. So now, you probably all know, and I think someone was mentioning, you were mentioning Hans Kerstedt earlier, there's really good evidence that if you take a stem cell and you transplant it acutely after spinal cord injury in rodents, you get improved function. And you've probably seen it in the news. Geron's conducting a clinical trial with regards to embryonic stem cells. But a lot of people don't know that it's been shown with embryonic stem cells, it's been shown with adult neural stem cells, fetal precursor cells, bone marrow-derived cells, and others. This is not the first. There's been a lot of stem cells people have injected into the mammalian, the rodent spinal cord, and gotten improvement and recovery. So the question is, what clinical trial do you do? I don't know. But the thing that you don't hear a lot about, which is very true, and it's important for all of us, is that these approaches don't work in the subacute or chronic period. So they just don't work well when you transplant the cells later. So the question is, why is that? Why would it work early and not late?
May I ask what you're defining as early?
So in a rodent, it's about 10, we got to get in there before about 10 days. So that's right. So the scar tissue is probably already formed, and I'm sorry?
Can't you remove the scar tissue?
That's a great question. Every time you remove scar, what do you get? Another scar. Yeah. So surgeons have tried that. They call that freshening the lesion and so forth. There are some classic experiments that people have done that.
What's the experience with the steroids?
Well, steroids block inflammation, and they help survival of cells. They decrease the inflammation, and they can reduce scarring, but they don't prevent scarring. But they can reduce it to a degree. All right. So you've got. I mean, we've pretty much finished the talk. You guys you got all the answers. That's really good. So so let me show you. I think the next thing is a practical experiment. So we wanted to think about this practically. You know, what are some of the molecules that regulate this? I think it is important that there's something about the mammalian response to injury that there's a time sequence in which scar evolves. And if you get cells in there during that period, you're going to get more scar cells. But the question is that long-lasting, and can we change it in any way? So if you look at this, if you think about it diagrammatically, and irregardless of lesion, if you imagine if you just cut the top of the spinal cord, so the gray area is an indication where you might cut the spinal cord, the red area is where a scar is going to form, and then this is the spared area of the spinal cord. So this zone here, as I told you, is going to become a cystic zone in humans. It's just a void, essentially, an inflammatory void where you don't get tissue regeneration. And that's a dead zone. Yeah, it becomes fibrotic even. So that zone, you know, unlike the salamander, we want to regrow that. We want to fill it in with neural tissue. That's what we want to do and bridge it. So we know that there are, I'm telling you some things that I won't have time to prove to you, but we know that there are factors that push stem cells that, you see the animation one time, force stem cells to migrate up into the scar zone. No question. A lot of investigators have shown that there's factors that pull the stem cells towards where the scar is going to be. And our lab and some others have shown that they participate in scar formation. But the question we asked as a lab, and we're going to get to a practical neurobiology experiment to finish the lecture and show you, I think, some of the practical issues with transplantation. One question you might ask is, what is it, or is there a force that pushes stem cells away from that cystic zone, right? Because if you've got cells that are being drawn into the scar, why don't they just march right into the cystic zone like the axolotl cells do and fill it in? So we hypothesize as a lab that there must be some repulsive cue. There has to be something that pushes out the stem cells. And you may say, well, Phil, is this really that important for transplantation? And this, I think, is pretty good evidence of it. So this is a study done by Brian Cummings at University of California, Irvine. And what they did is they took human fetal stem cells, and they transplanted them into the spinal cord injured rodent, and asked where do they end up and what do they make. Okay? And so the typical experiment, nobody grafts their stem cells at day 0 because it's impractical. But if you do that, in other words, therapeutically it's impractical. But if you do it in a rodent, what happens is all the cells you see here, the brown, I should say, all the brown cells are the cells they transplanted. And you see what happens at day 0, they get caught in what's going to become this cystic void, and they hate it there. They don't differentiate. They stay very immature. Most of them die. So nobody transplants at this time point. It's just a very bad environment. If you graft 9 days after injury, and sort of the golden period, people graft a few days up to about 9 or 10 days after injury. If you do that, they avoid this cystic zone, and they migrate all up and down the spinal cord. And whenever people have done that, they get repair. They get improvement. And it doesn't matter what kind of stem cell you put in. Some do work better than others, but many stem cells improve function. So this tells you at that little period, there's a beneficial environment to get the cells to distribute and do whatever they're going to do. But if you do that same experiment now a month after an injury, the cells now, I don't know if you can tell, but they're really focused in this scar zone, and they essentially contribute to the scar. And they lose their ability to produce neurons and other cell types. They only produce these scar-type of astrocytes. So this tells you that the environment changes, right, over time. First it sucks them in, then it pushes them out, and then it draws them back into the scar zone over time. So what might be, the last key thing here is, of course, you might note, is that none of the cells enter into the central void. So now we're going to ask the question, can we fool, so I talked about niches, talked about adult niches, and talked about the niche in your hippocampus and support cells and so forth. So I'm proposing to do, there's an injury niche, and that injury niche is stimulating the cells to become scar-forming cells. So the question is, can we fool, I like to call Mother Nature, can you fool Mother Nature, aka the niche? We need to fool that niche so we can stimulate regeneration. And I always like to make sure someone else has proven an experiment before I do it, because I'm not that smart. So I looked around, has anybody ever fooled Mother Nature? I wanted to see if anybody had ever done that. And it turns out that there was, in the 70s, the Chiffon butter company, they fooled Mother Nature. Do you remember that? But it had terrible consequences. And I remember the lightning shooting down and everything. When she discovered she was fooled, it was terrible consequences. But Chiffon can. So I figured if they can do it, the butter company, that we can do it too. Okay, and so this is the team that was going to try to do it. I don't know why it's yellow. Maybe we're afraid of Mother Nature, but this is Don Maris and Audrey Petit, two scientists in the lab, who set out to ask what is it that repels stem cells from the middle of the mammalian spinal cord. And this is the real neurobiology part of the lecture. So Audrey did an experiment where she took the embryonic nervous system. And so when you're in your early embryo, you're just a tube of neural tissue, just a tube, hollow tube. And that's what you see here. And anatomists call the top of it the roof plate, and they call the bottom of it the floor plate. It's pretty good. It's easy to remember. And so what Audrey did was she took this neural tube and she cut it open. And you see the dotted line there. She cuts the top of the neural tube and it falls open. And we call this the open book prep. It's the open book prep of the nervous system. And what Audrey wanted to do was put stem cells on there and ask, what do they become when they mingle with the environment of the developing nervous system? Can we make neurons and so forth that might be useful for spinal cord repair? And she couldn't get that to work. But along the way, she discovered something that seemed to be very fundamental. It happened over and over again. So in this case, the stem cells glow green. We've engineered our stem cells so they glow green. She puts them on the middle of the slice, and this is what happens over time. They line up in the middle. This is 12 hours later. They start to spread out at a day, and I hope you can all see it, but by 5 days, they form two highways, and they avoid the middle, which is the floor plate. Can you see that? So it's separated, and they void the floor plate. And we didn't know what that meant. We weren't smart enough to think ahead of what I'm going to show you, but I'm going to pretend we did. So we had the idea that maybe this had some relevance to spinal cord repair. So what possible relevance would it be? Pardon? They avoid the cut area, but this isn't actually the cut area, because we've cut the roof plate. And in fact, you're so smart, you have the next slide down, because it's not the cut area. Because if you do the reverse of this experiment, you turn it upside down, and now you cut the floor plate so that the roof plate's in the middle. They stay right there. They don't move off. So it has nothing to do with the cut. So it has to be some physical force. But we asked, is there a physical or molecular cue that might also be present in the injured cord? So the next thing Audrey did is make a list of what things are made, right, in this area of the embryo. She came up with a long list. And because we're supposed to be going home at some point, I'll tell you that what she did is she proved that none of these other factors were important, but this one factor, called netrin, seemed to be critical, essential. So what she did is she created mice that lack this molecule. This is a mouse that can't make this netrin. And then she made that neural tube. She cut it, did the open book prep. And now you can see, this is a mouse that is normal. And you can see there's that gap. See, they line up here and here, but they avoid the middle. This is a mouse that has some netrin, so one gene is knocked out and the other one's normal, and they still make that pattern. And this is a mouse that lacks both copies, so it has no ability to produce netrin. And now they line up along the floor plate and they're happy. They just sit down there. And what was exciting about this for us is that there's many inhibitory factors made at the floor plate. But the stem cells, these are adult neural stem cells, the only ones they cared about really was the netrin. So what we got excited about is that maybe there is just a key factor that's important in this process. She repeated this with embryonic stem cells and saw the same effect. So it seemed to be universal. Whether you're an embryonic stem cell or whether you're an adult neural stem cell, you don't like netrin 1, and it's a strong enough stimulus to get them to move away. And this is just the quantification of that. Okay, so the last couple slides, how does this translate? Does it translate? So while we were doing these studies, and to be honest, we had no idea. We weren't thinking about repulsion at the lesion core. But while we were doing these studies, we read a paper by Dr. Burley, who who probably didn't think about lesion cores either, but he was just doing some expression studies. He was asking what proteins are made after injury. And one of the proteins that he noticed that was made is called netrin-1. Now, in this experiment, all of the this is a knife lesion of the spinal cord. So he just puts a knife into the rodent spinal cord, makes a little cut, and longitudinal. This is longitudinal, and the white areas you see here is this molecule netrin-1. And we got very excited when we saw this. This is right where the cut is. Netrin-1 was concentrated in that zone, but not in the outer area where the scar and the cord is relatively normal. And so this, for us, kind of a light bulb kind of came on. I thought, well, maybe that's what's happening. Maybe what is occurring is that netrin-1 is being upregulated at the site of injury and pushing stem cells out of there. There's also these other inhibitors, but as I told you earlier, like slits and other ones, the adult stem cells and the embryonic stem cells didn't seem to be inhibited by them. So the blood stem cells that come in, so when you have an injury or a lesion, the invading immune system, the blood system, the hematopoietic system, is making this factor. And we think that there's a war going on. So when you have blood leakage after an injury, the immune system comes in and it sets up a territory. It begins to eat up the dead cells, and it makes a boundary. It says, this is the territory of the immune system, and stem cells have to stay over there because you can work on the spinal cord, but this is going to be the domain of the immune system, and it pushes the stem cells out. And the proof of that is in this experiment. It might be a little hard to see, and I apologize. I probably should have blown this up. So if these are our green stem cells, they're put now on a spinal cord that has, this is a normal, healthy spinal cord here, and the stem cells, this is just if you cut the spinal cord coronally this way and lay it in a dish and put the stem cells on top, they have no, just try to keep your eye on the top of the cord. So there's the surface of the cord. We put them there, and they just, normal spinal cord, not injured, they just hang out. They stick to the spinal cord, and they like it. If you do an injury, now red is a factor that's made after injury, and this dark blue. And you can see that unlike here, the cells now move down. See, they're here. They move away from the injury site. So they move en masse. They move away. And if you do the same experiment, but you take out that factor, you delete this netrin, the cells now spread out, and even spread out better than the controls. Here it is, and this is the surface of the spinal cord. You can see that they don't move away. They don't push down like you see this population. So we think this is really good evidence that you can use this factor, and it's only one part of the puzzle, but you can use this factor to control where stem cells will reside after you inject them, and maybe you can start getting some cell restoration. So this is, I do like to say, I mean, this is really the tip of the iceberg. I think there's probably other factors, and the thing I haven't told you that now, even though we get cells to linger within the middle of the spinal cord, they're not completely restoring new neurons there. So that's the next thing. There's still other work to be done, but this, we think, is the first example that, other than the chiffon company, that where we fooled Mother Nature. So. I'll leave you with some thoughts and take questions. This is, I think you should, if you are, there's hope, and if you are thinking that there will be therapies for spinal cord injury, I think you can derive a great deal of hope because lower species regenerate, and many of them do. They regenerate very complicated aspects of the nervous system, and they do it by activating stem cells. There's good evidence that our stem cells are similar. We also know, the good news I didn't tell you about, but our lab and many others have shown that stem cells survive fine to trauma. They love trauma. They survive, they divide, and they expand. So it's not like we're short on stem cells. I think that the other take-home is that the injured spinal cord, it creates a new niche which isn't necessarily restorative. It preserves the life of the organism, but it doesn't allow for plasticity, for enough plasticity for regeneration. So the solution as we move forward is to keep trying to understand what it is that controls the injury niche, and to try to elicit more robust regeneration. So all this work that I showed you requires people, and it takes a lot to go from here to here. And this is some of the folks from the lab. And I'd like to thank some of my sponsors, one of them who's here in the room today. But this work is funded by the National Institute of Neurological Disorders and Stroke. We received money from the Paralyzed Veterans of America, the Paralysis Project, the Utley Foundation, the Nielsen Foundation, Christopher and Dana Reeve Foundation, and Frank and Penny Webster. And Penny Webster is here today. So we thank you very much for that support. And thank you for listening, and I'll take any questions you may have.
So when do you think it's going to be available to the public?
Well, so he's asking, when would this be available to the public?
When? Kind of a timeline.
I think that for chronic treatment of chronic injury, we're going to have these co-therapies. We're going to put in stem cells with other things. We're going to have, we may be immune suppressing, but we may be putting in things that digest up the matrix, digest scar. We may be blocking netrin, for example. I think that for chronic, it's going to be a while for these things to hit, because we're just now, if you know, we're really just conducting the very first embryonic stem cell safety trial this summer. And so we've got a long way to go from that point. So I think the technology, though, the pace of technology is moving very, very rapidly. People are really starting to dissect this apart. And so my prediction is that once the safety trials get done, people get an understanding of, we don't even know exactly how to inject the cells in there, how many to put in, and whether immune suppression is important or not, so forth. All those things are going to have to be worked out anyway, and that's going to take, I don't know, at least 5-10 years probably to really work out. The good news is that while they're working that out, these strategies are going to get really optimized in model systems, so that the design of the real new trials around the road in 5 years or so probably will include these technologies and be much more potent. So.
If you were a person with an injury, what would your strategy be?
Right now?
Yeah.
Right now would be, the best physical therapy that I could get. I would keep up that. I think patients, I'm not a doctor, I should say that, and I could defer to a doctor. But, if it was me, based on my understanding, there's great evidence that physical therapy is really good for bowel and bladder function, and also some return of, sometimes in some cases can be some return of function. I'd be having to that. I think the best candidate patients for clinical trials are going to be patients that are in good shape and in the chronic condition that maintain the wiring and structure of their cord by keeping active. So that's my recommendation. I don't see anything out. There's no clinical trial in the world that I would enroll myself in or or my sibling, because I don't think there's anything there that's ready that would be safe and efficacious personally. So. Does that answer your question?
Yeah, there are so many labs, it seems, around the United States and around the world that are closing in on various pieces of the puzzle. It's really hard for people in the advocate community and the patient community to understand what to even pay attention to.
Yeah.
I wasn't aware of the work that you're doing until tonight, so. And I live here.
Well, I think it is okay. It's difficult it's even difficult for me to keep up on everything. The problem with spinal cord injury is that every injury is different, and I really believe that there's going to be universal trials, but this is really going to be in somewhat a case of tailored medicine, because some people have more sensory deprivation or sensory injury damage than motor. It's asymmetric, different levels, and I'm leaving out some important complexities, but neurons in one part of the spinal cord are different than they are in the other, and so when you think about regenerative therapies, it's not going to be simple. There may be some cell-based therapies that might be universally applied, but we just don't know what those are yet, and it may be more difficult than that in terms of how do we tailor medicine. So what do you pay attention to? I think if you see something, in the press or in the literature, you have to really take into account, and we talked about it earlier, about what species did it do that in, what kind of injury was it, when did they put the cells in, was it acute or chronic, were they trying to remyelinate? So some patients there's good evidence, some patients have conduction problems, but some patients there's no evidence of slowed conduction, depending upon the physiology and if they've had the benefit of getting a physiological analysis. So every trial probably that you see, or every preclinical model that you should see, I should say, is really designed to test a very specific strategy, and that often gets glossed over, in the press, when they say the rats are walking. And I think you have to look very carefully at what people are doing and what they're targeting, how they modeled the injury, and what cell they put in, and whether every patient is going to be a little bit different candidate for different therapies. it's the best I can answer that, I think. So
When you talk about the scar tissue blockage, is there anyone looking at with incomplete injuries where you have I don't know, the communication between the whole cord in certain tracks?
right.
If you were to inject stem cells into the functioning part, would it grow the functioning part, or does the scar just still get in the way?
So for incomplete, that's a great question. So for incomplete injuries, the stem cell therapy in the Geron trial, for example, they're not designed to stimulate like regeneration of these neural processes over long distances. That trial is really designed to replace the insulation, replace the myelin. And as I was saying, some patients, there's evidence for conduction slowing in the spared area, and that's what those are targeting. Okay. There are people who are now working on, trying to stimulate plasticity, localized sprouting of nerves that are spared, and there's good evidence that could be a potent strategy because if you have, there's a classic paper that showed a long time ago, at least in rodents, that you only need as little as 3% of a motor pathway to be working and to get some stepping, and humans it might be higher, but it's probably we're talking like hundreds of fibers might be enough to change the modality of just some leg movement to actually some stepping movement. So the bar is for stimulating the spared pathway might be a lot lower, right? Just getting it to strengthen the connections that are there, and people are working on that.
Compensatory strategies, yeah.
Mm-hmm.
Speaking of the Geron trial, do you have a feeling or an opinion? I've heard mixed things about the scientific community and whether this is the right trial to do and so forth and so on.
Well, is it the right trial? I think that it's really important. We have to do safety trials. And so if you were going to design a safety trial, this is probably a decent safety trial. It's not at all going to reveal whether there's efficacy because it's going to be in clinically complete patients, and they're going to inject the cells, below, and they're really just going to look to see if there's teratoma formation and how if the cells are safe. I think those studies need to be done. So because I do think therapies are coming along, and so if we don't, and if you, for example, a Geron trial, a good thing to know is that it took them long. They had to design the injection device of getting the cells in the spine. It was a very complicated device they came up with because nobody had really done it. You just have to be complete. They have to be anatomically, clinically complete. That will be the patient population. So it's purely a safety trial because there's. Pardon me? Yeah. Mm-hmm. But it's a safety trial. If it was me, I wouldn't be in a rush to be in this trial because there's I cannot think of any reason why this would benefit the patient because you're anatomically complete, and as we mentioned, that the strategy here is really to replace the insulation. But if the insulation isn't going across the gap, there's no reason to replace it. So there might be some, who knows, maybe there's some benefit that could be, but I think it would be very unusual. So it's really a safety trial. We have to do those. Some of the first trials that were done in the U.S. Were done with fetal stem cells. Were done in syringomyelia. They were done in Florida, and we learned a lot. We learned about how to image the cells and how to get cells in there. And so if you compare and contrast spinal cord injury in terms of the number of clinical trials, it's devastatingly low compared to stroke or something like that. And one of the reasons for that is what we talked about, that every injury is a little bit different. And the other thing is that we just don't have in the U.S. really a good infrastructure for people trying to inject cells into the spinal cord. If that's our therapy target, we just don't have neurosurgeons and so forth trained and practical equipment to do that.
I have two questions. One is I guess a month or 6 weeks ago, President Obama signed the Christopher and Dana Reeve Paralysis Act, which in part is going to set up a North American clinical trials network. So you spoke to that. How do you see that happening? Is that something that you get involved in, or are you?
Well, I mean, not probably me personally. I think that it's desperately needed, and I think it's disappointing that we don't have more clinical trials in spinal cord. So the Christopher and Dana Reeve Foundation they talked about this for years, that we need to establish this, and so it's exciting that this has now been enacted, because it's if you want to do a trial in stroke, I could probably get a trial in stroke started by some of the research I have in my lab, because the infrastructure's there. People, the hospitals will just sign up for it, but we're not at that level, and that's what this is going to bring, I think. And why is that important? That's really important, because when you do these trials, you don't want to. I mean, there's an emotional side of this where people want therapies, and they want whatever they can get as quick as they can get. They want to have a potential for improvement. But the practical side is we need to do these experiments, or I should say these therapies, clinical trial, in which way which we learn, so that we'll get better and better at it. And the only way you can do that is by having multiple centers to control for safety and efficacy in the same way, and share data. And that's what's going to happen with the trials network.
Well, I also am curious about, you talked about the glial scar and all that. I'm curious about strategies to break that down. Mm-hmm. If you're involved with that, or
yeah.
I know I've heard Stephen Davies in Colorado talk about Decarin and other things. Can you just speak to a few of those strategies and what's happening there?
So we've taken a cellular approach. We're trying to change the cell's attitude. So our approach has been to try to make these glial cells behave more like embryonic glial cells, so where you don't get a scar. There's very interesting work where people are using bacterial enzymes which naturally degrade some of the proteins that are found in the scar, like chondroitinase, and that is proving to be potentially effective. And what essentially this does is they inject an enzyme into the spinal cord, and it eats up the protein. So I showed some of them here, but I didn't talk about them. But these proteins are highly. So one of the problems with the scar is that some of the things that are made there are highly charged, and the charge of those proteins, the regenerating nerves don't like. They're repelled by that charge. And so if you cut off the ends of these proteins, then you can create an environment where the charge is appropriate and the axon can grow. And there's, you mentioned Stephen Davies, James Fawcett, Jerry Silver. There's several labs that have used an approach with different enzymes to degrade the scar, the proteins in the scar, and they get growth. And there's been some really remarkable effects of this. Now, it's mostly been in the sensory side, and sensory regeneration of sensory nerves. It hasn't been effective with motor nerves, and one of the reasons for that might be that motor nerves just aren't, whatever their genetic program isn't as well designed to regrow. But so far, people have tried freshening the lesion, like resectioning it. It doesn't work very well. We're trying blocking blood components to prevent the scar from forming, stimulating the cells to be less scar-like, and you can, it looks like there's evidence that you can digest these proteins too.
So due to time, we'll have to conclude with that question. I'd like us all to thank Dr. Horner for presenting tonight.